Good afternoon, everybody. So first of all, I want to thank Dr. Calvin Chu and Laura for inviting me. I think it's always very exciting to talk about research. And like Evan said, it's an important part. I think a lot of the things that have been discussed here today in terms of symptoms, hopefully I can explain a little bit about what is happening in the brain. First, one thing, the chair of my department always makes me do that, so I have no commercial interest or conflict of interest. I'm trying not to sell any commercial products to you. So I'm from the Department of Radiology, and so we do a lot of brain imaging, also known as neuroimaging. So a lot of you might be familiar with MRI or CT scans. So with an MRI scan or a CT scan, we can typically show brain tissue and the anatomy of the brain, and we use those scans. What we're really interested in, what our major part of the business is it's called positron emission tomography, PET scans, and that is a nuclear medicine technique. And we use that to look at neurotransmitters in the brain. And the neurotransmitters, you might have heard that already before today, those are sort of the chemical messengers in the brain. We can look at other things in the brain as well. We can look at metabolic demand, and we also can look at molecular changes in the brain. So for our research, and our research particularly in Parkinson's disease, we use both techniques in a complementary fashion to see what is going on in the brain of Parkinson's disease patients. So the one question is, well, why do you want to know that? So a lot of the knowledge that we have about Parkinson's disease, especially in the old days, we actually know from post-mortem studies. And what does post-mortem means? After death. So you need to die first before we can take your brain out and start figuring out what was going on in the brain. So short of killing our research subjects and looking at the brain, we came up with the imaging techniques. The other disadvantage of post-mortem research and the great advantage of imaging techniques is actually that with post-mortem disease, typically patients die at the end of the disease. So you're only looking at what happens in the brain at the very, very late stages of the disease. You don't know what happened at the early stages of the disease or the mid stages of disease and how that affects your symptoms. So with these imaging techniques, with these what are called in vivo, within the living imaging techniques, we are actually able to look at earlier stages of the disease. And so if we understand these earlier stages of the disease, what is going on early on, we might actually provide a better understanding of the symptoms that may occur in Parkinson's disease that are going on early on in the disease. If we understand what is happening in the brain, and if we can correlate that with symptoms of the disease, then we actually might go to the pharmaceutical industry and we say, well, symptom X is related with these and these changes in the brain, and maybe you can come up with some kind of medication that might help. So with the PET imaging technique that we use, and this is just to give you a little bit of a background so that I can explain the next slides better to you, we look at, like I mentioned before, at these neurotransmitters in the brain. So how do you do that? Well, you're interested in what they call a molecular target. For example, we all know that the loss of dopamine is very important. So you look for a chemical or some kinds of substitute or something that likes these dopamine neurotransmitters a lot, that can bind to it. You make that target, you make it radioactive, you synthesize it, and then you inject it in the person. That's where the nuclear part of it takes place. You have annihilation, and it all doesn't matter. The most important part is you then go into a PET scanner where we get pictures like these. So this is a typical PET image of somebody's brain. In this case, the molecular target that we used was glucose. So your brain, the source of energy for your brain is glucose, sugar. So if you want to look at the metabolic demands of your brain, you inject sugar, you make it radioactive, you go through all the processes that I just showed you before, and you get a picture like this. So how would we interpret a picture like this? So you're looking, oh, yeah, the laser. So you're looking at the brain. There it is. You're looking at the brain, and you see all these red areas here. Those are called hot areas. Hot areas means there's a lot of radioactivity emitting from that. Thus, it means there's a lot of activity, there's a lot of sugar going to those areas. So you have these red areas, and then you have these blue areas. There's very little neurons that have a metabolic demand, so there's not a lot of activity. So this is what you're looking at, is a very healthy brain of somebody who has a high metabolic demand. The important thing to remember from this picture is hot means, or the red color means there's a lot of activity going on. Blue or cold means there's not a lot of activity going on. So you heard me talking about nuclear medicine and radiation and all that kind of stuff. One point I want to bring up, it's very safe. The amount of exposure you get exposed to is very low. Oftentimes, it's a multiple of what you just get exposed, just standing here. You have a certain exposure for just being alive and walking around in this planet. If you go up in an airplane, actually radiation increases. All the amounts of radiation that we use are well below that. So even a regular CT scan that you might get at the hospital probably has more radiation than we have here. So I don't want to scare you off by talking about radiation. Everybody thinks that we're talking about a nuclear reactor. It's very low amounts. And I think this is in your handout as well. There's an organization called the SNMMI, and they have a lot of background information on that as well. So this was the little background on what we do on the PET imaging. So what are we interested in? So Parkinson's disease is actually a disease where Lewy bodies present related to alpha-synuclein. I don't know if anybody might have mentioned that before, but that is the main pathology. Unfortunately, there's not a radio tracer that we can immediately target these Lewy bodies, so we cannot immediately visualize it. Although the Michael J. Fox Foundation just gave $1 million reward for the investigator that actually can do that. Won't be me, I can tell you that. We can see the end result of that, and everybody knows the end result, and that is the loss of dopamine. So at the University of Michigan, we actually have a dopamine tracer. We actually invented that dopamine tracer, and that was done by Dr. Kirk Fry here. And that tracer is called DTBZ. And it's a very well-established tracer for which we can, with which we can visualize dopamine in the brain. So what does it look like? Again, remember, red means a lot of activity. White, actually, it's red hot. It means even more activity, and blue doesn't mean too much activity. So if you look at a normal brain, then we look. This is the striatum, and so most people might have heard of it, the putamen, the caudate nucleus. And so that is what is affected in Parkinson's disease. And so if you look at somebody who doesn't have Parkinson's disease, you see that here, there's a lot, it's even, it's red hot, it's white, so there's a lot of activity going on. So that means that there's a lot of dopamine in the striatum. If you look at somebody who has mild Parkinson's disease, you see actually two things. That all that white has disappeared, so it's not red hot anymore. It's asymmetrical. One side is actually affected more than the other side. a lot of Parkinson's disease patients have an asymmetrical presentation of symptoms as well, which might be explained by this. And if you look at the back portions of it, where the putamen is, there's actually more loss than the more frontal portions of the striatum, where there is still relatively a lot of activity. We know that people with Parkinson's disease, who we all now know that there's a lot of non-motor symptoms that present very early before you get the diagnosis from the neurologist that you might have Parkinson's disease. But at the time of diagnosis, so neurologist tells you, you've been diagnosed with Parkinson's disease, we know that about 60 to 80% of the dopamine neurons are already gone. So the loss that you see here is already incredible, 60 to 80%. Having said that, it's also a testament to the brain that you lose 60 to 80% of a neurotransmitter and you still function relatively well up to a certain point. Now with disease progression, I should say this is not the same person, so it looks like it's a little bit more activity here. But with disease progression from moderate PD to moderate severe and very severe Parkinson's disease, all the dopaminergic activity is just gone. You can see here there's very little red activity going on, so very little dopamine there. So what do? You lose dopamine, so you get dopamine replacement therapy. Dr. Ritter had a very nice slide, and he should recognize this slide because I have the same, same slide. So we know people. We have people in common. And so I'll approach it from a little bit of a different perspective. So he sort of laid out what the motor and non-motor symptoms are. I'll tell you what the dopaminergic responsive and non-responsive symptoms are. And so for the motor, maybe slowness of movement, stiffness, tremor, perhaps to a variable degree only, respond relatively well to dopamine replacement therapy. But for balance problems and gait problems, dopamine doesn't respond. There's not such a good response to dopamine. It doesn't really make your balance or gait problems any better. All these other non-motor symptoms that Dr. Ritter was talking about have a very poor dopaminergic response as well. So why is that? Well, and I think you might have heard that from many speakers before, Parkinson's disease is more than just losing dopamine alone. So this is all based on post-mortem studies, but a famous pathologist, Heiko Braak from Germany, actually sort of staged the progression of Parkinson's disease. And he proposed that it's actually an ascending disease. So it starts off with pathology developing really low in the brain stem areas, where some of the non-motor symptoms might be explained, such as REM sleep behavior disorder or others, and then slowly move up till you only hit stage 3. That's actually the stage where you then have loss of dopamine in the striatum and the substantia nigra. So what we initially considered to be classical Parkinson's, and we all thought, okay, that's the substantia nigra and that's the connected striatum where all the pathology takes place. There's already perhaps 2 stages before that, and then it sort of ascends up. With all of these different stages of pathology developing, you hit different neurotransmitter systems. So that's why it's not dopamine loss alone, because in the end, the disease is affecting other systems as well. So this is sort of what I'm referring to. This is a very famous slide. Every self-respecting researcher uses this slide. This is by Langston, and he calls it the Parkinson's complex. And with the loss of dopamine in the substantia nigra, what I just mentioned is stage 3 of this ascending disease, we're only seeing the tip of the iceberg. There's so much more going on. And so that's what we're trying to focus on with our studies. And in particular, we are looking at acetylcholine. So acetylcholine is another neurotransmitter that you can find in the brain. And it creates these typical pictures of somebody who has a normal distribution of acetylcholine and a Parkinson's patients where there's definitely a reduction and where there's not as much redness in that picture. Having said that, not everybody with Parkinson's disease has cholinergic loss. So we've done some studies, and there are plenty of people who are kind of in the normal range of it, and it's up to about 60% or even higher that are in the normal range. So they have the same amount of acetylcholine in the brain as somebody who doesn't have the disease. There's different regions of cholinergic loss. Either your brainstem and thalamus can be affected versus more your cortex and the more frontal regions. And so there's a differential pattern where some people have it more frontal, some people have it frontal and thalamus, and very few select have only thalamus. So having said that, the message is not everybody has that cholinergic loss. However, the degree of cholinergic loss actually does associate with cognitive problems. And I think Dr. Ritter mentioned that as well. If you have cognitive problems, you actually get prescribed cholinergic medications. So we show that here in research as well, that the loss of acetylcholine has to do with cognitive loss. We took our research further. We also looked at the balance and gait problems. And so I think in one of the very first studies, what we really showed is that people who frequently fall or have a falling history actually have loss of acetylcholine in the brain as well. It's typically associated with the thalamus. It's a deeper structure inside the brain. It's connected to the brain stem, but also with the higher up regions in the brain. So there's definitely a connection between cholinergic loss and falling. There's also a correlation between cholinergic loss and gait speed. And it's actually somewhat interesting because what we saw is that the people who have cholinergic loss have a decreased gait speed, the Parkinson's patients. But interesting, the Parkinson's patients who don't have cholinergic loss, but they do have dopaminergic loss, because every Parkinson's patient has dopaminergic loss, don't have a decrease in gait speed. It's slightly decreased, but it's very comparable to gait speed in people who don't have Parkinson's disease at all. So cholinergic system really might be related to falling, we already saw that, and to gait, ambulating through your environment. We also looked at balance. We have a fantastic device at our lab where we can measure your balance in all kinds of different conditions. And what we saw is that the sensory processing that is associated with balancing, so when you balance, you get all kinds of inputs from the different systems. Sensory processing is affected by the cholinergic system as well, and you don't do it as well when there is cholinergic loss, and there's actually a very nice linear relationship with that. REM sleep behavior disorder, I think there was a whole talk about it. That's true, right? There was a talk about it this morning, so I don't need to go into detail what it is, but Dr. Vikas Kodegal, who some of you might know, actually worked on a study in our lab, and he showed that REM sleep behavior disorder is also associated with cholinergic loss. Coincidentally, REM sleep behavior disorder is also associated with cognitive problems. What is also associated with cognitive problems is balance and gait impairments. So it's all connected, and these are all cholinergic mechanisms. I don't think I have a whole lot of time, so about five more minutes. So I'll keep this very briefly. I had a lot of slides on that. Like I said, there's more information in your handout, because there's one other thing I want to emphasize after this. So we also look at beta amyloid. Beta amyloid is the plaques. In Alzheimer's disease, you have the so-called plaques and tangles. beta amyloid are the so-called plaques. And they also appear as a normal process of aging. And so we were looking at what that might mean for Parkinson's symptoms. We have tracers for that as well. And like I said, I'll briefly go through it. But the most important message is, if you look at this beta amyloid, these plaques that you see in the brain, they are associated with balance problems as well, and with cognitive problems as well. This is actually part of a larger message that I'm going to try to convey in the next few slides. So Parkinson's disease, symptom disease severity, is not just the degree of dopaminergic loss. We saw that, so I already showed you that acetylcholine actually plays a role in that as well. So if you have Parkinson's disease, loss of dopamine plus loss of acetylcholine, then you might have falls and gait and balance problems and cognitive problems. If you have Parkinson's disease and you have these Alzheimer's amyloid kind of plaques, which is, it's not that common, I have to say that, it's not that common. But even to a very minor degree, that might just be normally age-related, you have gait and balance problems, and you develop some cognitive problems with that as well. Then the next step is actually hypertension. So that's actually, you know, a modifiable risk factor. We look at MRIs, and what we can look at in MRIs is the so-called white matter. White matter is sort of the highway, it's the connections between the different cells in your brain. I always use Michigan as an example, except for around where I live, the roads are very good right now. But if you have a lot of potholes in the brain, in the road, you know, your commute or your travels will not be that fast. Well, it's the same for the highway of the brain. If there's a lot of damage in that, which can be caused which can be caused by hypertension, high blood pressure, then you get these damage in the connections of the brain as well, and those will appear as these white spots that you see here in the brain. So that's a modifiable risk factor. Actually, you can treat hypertension, and it would be very good to do because that has been associated with gait and balance problems as well. Diabetes, and this is all research by Dr. Vico Skodegal and by Dr. Meir Petru does some research in that as well. So diabetes, up to the, especially type 2 diabetes, up to a certain degree, is also controllable. You know, and you would want to control it because, again, if you look at the MRI in the brain, in this case, we actually looked at the cells in the brain, the neurons in the brain, and we actually see that with diabetes, there's an associated loss of cells of brain volume. Again, that might lead to balance and cognitive problems. So the latter two, diabetes and hypertension, are modifiable risk factors. And so the overall conclusion and the overall message I want to give is, especially in the past, when we did Parkinson's research, we spoke a lot about the loss of dopamine alone. I think for the last 10, 15 years, there's really a paradigm shift going on where we speak about all these other things. And every single little thing that you get on top of your Parkinson's disease, on top of your dopaminergic loss, is not going to help. And I think that is what we're showing with our research. You have the loss of acetylcholine. There's nothing much you can do about that. Amyloid plaques might accumulate. We don't know if we can do something about it or not. There's some promising studies out there. Hypertension might lead to damage in your brain, in the connections in your brain. We can treat hypertension. Diabetes, connectivity problems in your brain, brain volume loss, it can be brought under control. So whatever you can do to control these modifiable risk factors, you definitely should do. And because it will, in the end, it definitely will help you with your balance and gait problems. But I think that, and I hope that our research shows that it's not only dopaminergic loss, that we also need to really venture into these other areas, and that we need to come up with good medications. I know, you know, if you have cognitive problems, the doctors prescribe the cholinergic medications, as I just mentioned. The reality is the cholinergic medications that we have right now are okay, but are not great. So, hopefully we'll get to some better medications, and hopefully at some point we can start doing clinical trials where we look and can see if these cholinergic medications also can help with falls and balance and gait problems, and not only with cognitive problems. So I sort of want to leave it with that. Dr. Chu mentioned we do have the Udall Center for Parkinson's Disease Research at the University of Michigan. We are also sponsored by the VA and the Michael J. Fox Foundation. And this is our lab. I'm the bald guy, in case you didn't recognize me. And Dr. Bonin is the director of the lab. I'm the co-director, and we do a lot of research there. And I'll leave it at that. I think there was one there. Oh.
Are there any biochemical markers that are related to like the extracellular matrix that you can identify and do some research on?
So, what are you thinking about specifically?
I'm thinking about the...
well, I mean, when you talk about the extracellular space, for example, beta amyloid is extracellular, right? It sort of connects, it sits between the cells. So we have a marker for that. It's not the most important thing in Parkinson's disease. In dementia with Lewy bodies, Alzheimer type kind of plaques are much more important. So we can look at that. If you talk about extracellular, we're also talking about the white matter, that connectivity and those white spots you saw in the MRI brain. So that's the extracellular space. In particular, if you talk about neurotransmitters, then you're looking at receptors and transporters and all that kind of stuff.
Okay, so thank you.
Is it not quite satisfactory? Or?
It's a start. Thank you.
No, I mean, but what specifically are you after?
There's like some specific protein biomarkers that are out there that you see that can identify or be associated with what's happening?
Well, I mean, like I mentioned in the beginning, the great quest is for the Lewy bodies. So what we see in Parkinson's disease, what happens in the substantia nigra has these dopamine-producing neurons. And what we know is that Lewy bodies are these little inclusions start developing in these neurons. And in the end, those neurons die off, don't produce dopamine anymore, that goes to the striatum. So that is the great quest. That is really what we want to discover. Well, we know that the Lewy bodies are there, but what we want to have is a tracer to look at those Lewy bodies. And that we don't have. Like I said, you can win $1 million with the Michael J. Fox Foundation, but it indicates, by the way, that the Michael J. Fox Foundation is very serious about it. I mean, they put on a big price for that, and but that's the big quest. We don't have it yet.
People are working.
Can you tell us a little bit more about the acetylcholinergic? I can't even say that word.
Yes.
The medications that are used to treat that loss?
Yeah, I'm not quite an expert on it, but what you have are the acetylcholinesterase inhibitors, like donepezil is one. It's known as Aricept, and
Donepezil is Aricept.
And
ribostigmine is Exelon.
Oh, Exelon, yeah, that's right. And I think those are the two major ones, right, that you would prescribe, right? So that.
Excellent.
So I'll leave it up to the doctor.
So the medications that are used to treat the cholinergic, the acetylcholine system, the brand names are Aricept, ARICEPT, and Exelon, EXELON. These are medications that are approved for the treatment of cognitive impairment and dementia in Parkinson's disease. So they're prescribed when people start to have problems with their thinking and memory, but they are not approved for any other reason. So we don't use them to treat falls or any of these other problems. There are some small studies, and actually part of the Udall grant is looking at whether or not these medications can actually improve gait and falls in Parkinson's disease. But it's not something that we as physicians routinely prescribe for falling problems. It's not clear yet that it actually helps. So just in the interest of time, one more question.
Is there a way, method to increase the amount of acetylcholine in the brain, this red activity for Parkinson's patients?
So again, that would be those medications, the acetylcholinesterase inhibitors, that is their function. So when acetylcholine gets released into the brain by the neurons, as part of the normal process, you have enzymes that then break it down, goes back into the cell and sort of gets recycled. And what you do with these acetylcholinesterase inhibitors, you inhibit that activity, so you just make sure they don't get broken down that easily or that quickly, so that there's more available.
The inhibitors you're talking about, the anticholinergic inhibitors, is that right?
Yeah, it's a little bit complicated. It's acetylcholinesterase, and that is the enzyme that breaks down your regular acetylcholine.
Right, but I'm concerned, for balance. I feel that very strongly that when I stand. I don't have the sense of the perception of left and right, and that I'm steady.
Right.
And that's the absence of acetylcholine, as
Right.
I understand you saying.
Right, so one of the studies that I did is indeed that we looked at sensory processing of standing
Right.
balance,
But
and that was related to acetylcholine.
is I'm looking for the impossible, I guess. I want to increase. I want that sense of balance back.
So I'm sorry, I'm gonna stop, because this seems to be a very personal question, so related specifically to you. There are actually a lot of reasons why someone might have balance problems, not all related to acetylcholine in the brain. So you may have other reasons why you have balance problems. It's not just Parkinson's and not just acetylcholine deficiency. But there are no ways to add acetylcholine to your medication regimen to improve that. So I'm sorry about cutting off the questions and the time, but we are actually running late, and I wanted to give enough time to our last speaker.